Assalamualaikum to Madam Saidah and to all my friends. Today we are from Group A for AS12003 will presenting our study case BIO270. Alright, before we start, let me introduce my group members first. The first one is Nurul Nandia binti Muhammad Arafi. The second one is Nur Ain Nabiha binti Muhammad Khairul Nizam. The third one is Farah Wahidah binti Sudin. And the last one is me, Nurul Ain Farisha binti Abdul Rahman. So our topic is rice plants, Oryza sativa, produce an important staple food. It is crucial that this plant can thrive against stress such as drought and salinity. I explain several strategies to improve such stress tolerance in rice plants by giving example. Rice is a staple food in daily human life, especially for Asians. Rice can be anti-cancer, treat diabetes, and liver diseases. Rice is a natural anti-inflammation and gluten-free. So there are over 40,000 different varieties of Asian rice, which are broken into 4 major subspecies. First is indica, japonica, aromatic, and glutinous. The 2 major subspecies are the long grain indica and the short grain japonica.
First, drought stress. We have 3 common types of drought-effect rice production, which are early water stress that cause a delay in seedling transplantation, mild sporadic stress having cumulative effects, and less stress affecting less maturing varieties. Drought stress induces various physiological and biochemical changes in rice at different developmental stages. It is thought that the ability of the plant to modify its roots to grow thicker and deeper into the soil might be an important mechanism to avoid drought stress, and there is ample evidence that assimilates are relocated to roots instead of shoots as a response to water stress. On the contrary, some research demonstrated that roots grow in rice decrease under drought stress. These findings show that the response of roots to water stress is highly dependent on the crop genotype and period and intensity of stress. The impact of drought stress on rice yield also depends on the growth stages, with mid-tillaring, flowering, and panicle initiation identified as the most sensitive stages. Next, salinity stress. Salinity has been a key abiotic constraint deficient crop production worldwide. Attempts in understanding such tolerance mechanisms has been revealed several key enzymes and altered biochemical pathways inferring resistance to crop plants against such stress. The past decades have witnessed intensive research in development of such tolerance cultivars via conventional means, improvised by modern era molecular tools and techniques. Rice, Oryza sativa L, is the staple food crop across several countries worldwide. Being a lycophyte by nature, its growth is severely impacted in presence of excess salt. It is because rice is susceptible to salinity, specifically at the early vegetative and later reproductive stages. Rice genotypes show wide variations in salinity tolerance due to additive change effects. Studies indicated that rice is more resistant at reproductive and grain filling than at germination and vegetative stages, as well as low levels of salinity can increase the resistance of rice to higher and later salinity levels. At present, salinity is the second type of stress and is the most predominant hindrance to rice production of the drought. The effects of the salinity on the growth and yield of rice in the field have been well studied, including the study of genotypic variants for salt tolerance among the paddy germplasm.
Okay, so what is the effect of soil stress on rice? The effect is impact root system establishment, leaf rolling, chlorosis, fewer tillers per plant, reduced biomass, shorter plant height, lower thousand grain weight, ultimately leading to reduction in harvest index and grain yield. Okay, for the next part is strategies to improve stress tolerance in rice plants. The first one is modulating root growth and development. Root development is deeply involved in plant tolerance to abiotic stress such as drought and salinity. Modulating root growth and development provides a potentially useful approach to improve plant abiotic stress tolerance. Okay, so there is three adaptive mechanism has been confirmed for rice under drought stress. The first one is osmotic adjustment in roots, if possible, in condition with a relatively small soil water reservoir. The second one is increased root penetration into the soil. The third one is increased root density and the root to shoot ratio. For example, by increasing the number of root or root hair can help rice plant absorb more water and nutrient from the soil, thereby improving their drought tolerance. So, the higher the root development, the higher the stress tolerance.
Alright, the next one is breeding. Breeding is another strategy to improve stress tolerance in rice plant. For example, by using several methods such as introduction, hybridization, and mutagenesis for generation of highly variable salt-tolerant breeding population, considerable success has been obtained in raising rice genotype with improved tolerance towards salinity. A number of genetic features are chosen during rice genetic improvement in order to boost yield potential, yield stability, and broad adaptability. Three primary factors determine the rice plant grain yield is the weight of the kernels, the number of spiculates per panicle, and the number of panicles per unit area. These factors vary in much, contribute to grain output, and depends on the cultivator genotype and environmental factor. On the other hand, the primary factor influencing rice grain yield might be plant architecture, which is tiller pattern, plant height, leaf form, and arrangement. For example, control the rice tiling. Rice tiling is characterized by pattern and angle which establish how much area the branch takes up. There are two different of vegetative tiling among grass species that have been domesticated to the cereal crops. The first one is axillary and the second one is tiller. When tiling occurs, side tiller that resemble the main clump in the height and formed by the axillary meristem, and while the axillary branch is come in nodes that have developed at the higher nodes of the clump create axillary branch from axillary buds. Alright, the next one is control the plant height in rice. It is because it's being a key component of plant architecture. Plant height is a significant agronomic characteristic that is closely related to the both of the harvest index and yield potential. Plant height is determined by the total number of elongated internodes and the length of each extended internodes. A plant rice typically has 4 to 6 elongated internodes, and environmental influence as well as the quantitative trait loci and immediate the plant's height. The next one is the control of leaf development in rice. It is because it is the primary photosynthetic organ and the key players in carbon fixation. Leaves are frequently in the most visible element of the plant. Certain leaf characteristics like quantity, shape, size, and thickness and the chloroplast level are crucial to determine the leaf. The plant's ability to create the zoomus and the drive. The genes that control the orientation of the three functioning leaves and development which major factor in the rice grain yields are briefly introduced in this session. The next one is hybridization. It has been established that the best way to increase crop yield potential for many species, particularly cross-pollinated ones like sorghum, cotton, and maize, is through hybrid breeding. Hybrid virgus or heterosis refer to a phenomenon which is F1 hybrid resulting from crossing between genetically diverse inbreed varieties perform better phenotypically from their parents. This phenomenon is utilized in hybrid breeding. Any biological process that functions better in the hybrid offspring may be a sign of superior performance. The next one is mutagenesis. This functional research and the creation of genetic variability can both benefit from mutation. To comprehend gene function, mutant can be analyzed using forward genetic, which goes from phenotype to gene, or reverse genetic, which goes from gene to phenotype. Higher plants, on the other hand, have very low spontaneous mutation rates. One key tactic to raise the frequency of mutation is mutagenesis. It makes functional genomic research and the creation of novel genotype possible.
The third one is genetic modification. Genetically modified rice are rice strains that have been genetically modified. Rice plants have been modified to increase micronutrients such as vitamin A, accelerate photosynthesis, tolerate herbicide, resist pests, increase grain size, generate nutrients, and increases stress tolerance. GM rice is one that had gene or genes from another species or rice variety introduced into its genetic makeup using modern biotechnology techniques. Okay, so golden rice is genetically modified to produce beta-carotene, which is not normally present in rice. Beta-carotene is converted into vitamin A when metabolized by the human body. Example of genetic modification is researchers have developed salt tolerance rice by introducing genes that regulate into ion transporters, which help the plant maintain ion homeostasis under salt stress.
The next one is application of growth regulators. It is negative effects of high temperature stress on rice were effectively mitigated by the use of PGR. When compared to nothing applied control NAC, the exogenous application of PGR significantly enhanced the growth and yields of both rice varieties. The application of vitamin E, glycine, betaine, and salicylic acid exogenously resulted in an increase in rice yield and enhanced the plant resistance to high temperature. When PGR were exogenously delivered, leaf gas exchange was shown to be better than the NAC. The application of PGR in rice has been shown to reduce internal CO2 concentration and respiration rates and membrane damage while increasing the photosynthetic. For example, regarding foliar application of growth regulating compounds, spraying 1% pink pigmented facultative methylotroph (PPFM) resulted in the heightening of plants and was comparable with 0.1 ppm application of brasinolide. Spraying of PPFM might have stimulated the growth and plant height by increasing the amount of auxin and cytokinin in rice. Increasing moisture stress significantly resulted in a decrease in plant height under control for foliar application growths, regulating compounds.
The next one is rice seed germination. Rice seed germination is a complicated phenomenon of biochemical and psychological change that triggers the embryo's activation. Grain yield production and total dry matter are impacted by the crucial state of seed germination. There is negative correlation between salt stress and seed germination, as seen by the delay in rice seed germination caused by salt stress. Drought stress severely affects the crucial plant processes. Drought stress amplifies the oxidative stress in plants. Seed priming is an effective technique to cope up with multiple abiotic stresses. Seed priming strengthens the antioxidative defense system through upregulation of antioxidants enzyme. Seed priming memory helps in transferring the stress memory to their progenies. So this is the example of rice seed germination. As an alternative, seed priming can serve as a cost-effective, environment-friendly, and pragmatic approach to address global food security through sustainable agriculture innovation. So this is the picture of germinating seeds.
The last one is rice physiology. The rice plant experiences several stages, which is vegetative, reproductive, and feeding phase. The starting of seed germination marks the beginning of the vegetative phase, which continues with the growth of seedling, tiller appearance, and multiplication, elongation, and expansion of shoot and roots. After the vegetative phase, the reproductive phase begins with the formation of spikelets and followed by the fertilization.
In conclusion, there are various stresses that cause rice production to be disrupted, namely drought stress and salinity stress. So we need to use some alternative so that we can improve stress tolerance in rice plants, which is modulating root growth and development, breeding, genetic modification, application of plant growth regulators, rice seeds germination, and rice physiology. In this way, it can help our country economically, for example, by less spending on rice imports, and can increase our income by exporting more rice to other countries. Thus, can reduce the rate of hunger in our country in addition to contributing good nutrition to the human body.